you very much to everyone at MS Research Exchange for inviting us to talk. So Paul and I are going to tag team this presentation. And so I'm going to give a little bit of background on why we're interested in bryostatin and in the pathways that we've been studying. And then Paul's going to go through the data from the preprint itself. And so I'll get started. And so this is collaborative work that Paul and I have been doing now for a number of years. We were kind of friends and colleagues together and in the same PhD lab. And so this goes back quite a while. This is a clinical audience, and so I won't spend a lot of time, but just to set the backdrop, this is a schematic of the clinical course and pathologic evolution of a typical patient with MS. In the middle here is a disability over time for your typical patient that starts out with relapsing remitting disease. And as most of you know, relapsing remitting MS starts out with these discrete episodes of inflammation where the peripheral immune system gets activated and targets a different area within the central nervous system to produce clinical relapses, after which there's some recovery that is sometimes all the way back to baseline and sometimes incomplete. And many patients with relapsing MS will then convert to a secondary progressive stage where they see progression of disability even in the absence of our conventional measures of inflammatory disease activity, meaning clinical relapses or new MRI lesions. MS, in a lot of ways, has been one of the biggest success stories in therapeutics development in neurology. We now have more than 20 FDA-approved therapies. They very convincingly change the long-term clinical course of the disease. But they also all do more or less the same thing in the sense that they generally target the peripheral immune system, and they prevent our conventional measures of inflammatory activity, meaning clinical relapses and new MRI lesions. They target the peripheral immune system, mostly the adaptive immune system, and they are most useful in the context of relapsing MS. What we don't have therapeutically in MS are any treatments that slow progression when it happens independent of our conventional inflammatory activity. We don't have any treatments that promote myelin repair and therefore promote recovery of function for people that have established disability. And this is really two of the major goals going forward of therapy development in MS. And the reason that current therapies don't target these processes is because the biological drivers of remyelination failure and progression are distinct from those that are associated with relapsing disease. And one of those big drivers of these pathologic processes is what some people call smoldering inflammation or compartmentalized inflammation, whereas relapsing MS is driven by a peripheral origin of inflammation that's infiltration largely of lymphocytes into the brain. These later stages of MS are driven by continued inflammation that's associated with microglia and macrophages, the innate immune system within the brain. But these cells are not just all good or bad. So microglia continue to be active throughout progressive MS. We see them at the edges of slowly expanding lesions. We think that they continue to play a role in progression. But they also have a number of adaptive functions that are critically important for repair. And so I've started to think of MS lesions like wounds, that can either heal or can fester depending on the evolution of the inflammatory response and the phenotype and function of microglia and macrophages within these chronic lesions plays a big role. And so we know that throughout the different stages of MS, these cells can continue to contribute to ongoing injury through antigen presentation, the release of a number of toxic molecules that cause ongoing demyelination or axonal injury. But we also know that that these cells are critically required for regeneration and repair. We know this from animal models. We know this from human studies and other diseases such as Alzheimer's. And they're required to phagocytose debris. So myelin debris is very potent inhibitor of remyelination. They secrete regenerative factors such as activin A, BDNF, NGF that promote repair, and they play a specific role in recruiting oligodendrocyte precursor cells and inducing their differentiation. And so a big therapeutic target are agents that can not just turn microglia and macrophages on and off in the brain, but ones that can actually move them away from maladaptive towards more adaptive functions. And this is some of the optimism around BTK inhibitors and agents that are targeting TREM2. And so with that background in mind, I'll kind of switch gears a little bit and talk a bit about how we became interested in protein kinase C or PKC and Bryostatin 1 in particular. So PKC is a kinase that comes in many different flavors. There's 10 different isoforms that are pretty broadly expressed across immune subsets. PKC, and these are not splice variants. They're actually present at distinct genetic loci within the cells. Two of the big classes of PKC are activated endogenously by diacylglycerol, which is produced downstream of phospholipase C in cells. And when diacylglycerol binds to PKC, it's activated. It moves from the cytosol to the plasma membrane, and that's followed by compensatory deactivation. And there have been a number of different drugs that have been discovered over the year that bind to this diacylglycerol binding site and can have different effects on PKC function, kind of like the way that rapamycin and tacrolimus have different effects on FKBP12 on downstream function. And so the 4-ball esters have been shown to be pro-tumorigenic. They're actually mitogenic in the context of the immune system. So PMA and ionomycin are used to activate T cells, whereas Bryostatin 1 tends to antagonize the effects of 4-ball esters. And because of differences in the conformation of binding, the interaction with plasma membrane, these different drugs can have different effects on PKC. And so we clued in on Bryostatin 1 kind of early on in this work. We were talking a little bit before the talk with Bob about how he became interested in Bryo 1, and we can talk a bit more afterwards. But we were originally interested because Paul had become interested in PKC and its role in neurodevelopment downstream of toll-like receptor signaling. And so he had discussed with me looking at this in the context of neuroinflammation because of the role of PKC in immunity. And so we started out in a, quite frankly, not very sophisticated way, looking at PKC modulators in the context of MS models. And with Bryostatin, we started out just by looking at its effects in EAE. So a number of you will be familiar with this model, which is a model of inflammatory demyelination, in which mice are immunized with a peptide that's derived from a myelin protein, MOG. And 1 to 2 weeks after immunization, they develop an autoimmune attack on myelin that's kind of centered in the spinal cord. And so they develop a progressive paralysis that can be scored clinically. And traditionally, you can look at treatment paradigms, both prophylactically, starting treatment at the time of immunization, versus starting treatment later after the immune priming has taken place to try to isolate different aspects of the therapeutic effect. Some of these models are monophasic, some of them involve relapse. And so we started looking at some of these drugs, including Bryostatin 1, in the C57 Black 6 model, which is a monophasic model of EAE. What we found was that Bryostatin 1 was very potent at attenuating EAE, which by itself, honestly, is not very exciting in my view. A lot of things work in EAE, but there were a number of aspects of what we found that got us more interested in this drug. One was just how potent it is. So in all these treatment paradigms, our dosing regimen based on prior studies that had been done with Bryo-1 and other models was 35 nanomoles per kilogram, so about 30 micrograms per kilogram, so 3 days per week. And when we deliver this drug prophylactically, so the green arrow here denotes when treatment was started. So when we treat mice at the same time of immunization, you essentially completely abrogate the onset of the disease, which is obviously about immune priming more than anything else. But we similarly saw a very potent effect when we delivered treatment after the onset of neurologic deficits. And we saw that this decreased the inflammatory response within the spinal cord, basically prevented the accumulation of CD4 cells. But the second thing that kind of got me in particular very interested is that we saw effects of the drug even at very late stages of the disease. So in this paradigm, we started treatment at day 28, which was kind of when these, after the peak of disease, when mice had plateaued and that initial wave of peripheral inflammation had largely subsided. And we saw that when we started treatment at that time, these mice were improving. And then when we stopped treatment, there was a rebound effect. So clearly there was some kind of purely anti-inflammatory action going on, but these mice never reached back to the previous baseline, which suggested some level of repair. The third thing that got us more interested in Bryostatin 1 was what the cellular target seemed to be. So when we looked at the effects of Bryo-1 directly on T cells, we really saw minimal effects. But we saw very profound effects on myeloid cells. And so this top panel here are dendritic cells from mice. And we found that at nanomolar concentrations, Bryostatin 1 not only prevented the release of classically pro-inflammatory cytokines like IL-12, but actually promoted the production of cytokines that are associated with a type 2 response or more of a tissue resolution response, such as IL-10. And we saw something similar in peripheral macrophages, where Bryo-1 at nanomolar concentrations was decreasing production of IL-12 and IL-6, but augmenting expression of arginase 1, which again is associated with a phenotype that we see more in the context of tissue resolution. And so the myeloid target, the potency, and the effects in late disease were originally what got us most interested in this drug. We next started doing some work with Paul Wender at Stanford, who's been studying Bryostatin for 30 plus years, to first figure out whether the effects of the drug that we were seeing were really dependent on protein kinase C. So because there's so many isoforms of PKC, it is a nightmare to try to look at this using genetic models. And so Paul Wender's group had produced a derivative of PKC that has a change in one of the side groups that prevents binding to protein kinase C. But then we also looked at a number of other either analogs of Bryostatin 1 called Bryologs, or structurally unrelated PKC modulators, such as prostratin and this derivative of prostratin, that had been shown to have more Bryostatin-like effects than 4-ball ester-like effects in other systems. And what we found was that in peripheral myeloid cells, the effects of Bryo-1 were dependent on PKC. So when you abrogate PKC binding, you lose the effect on IL-12 and IL-10 production, for instance. And you could replicate these effects with certain analogs of Bryostatin and with some structurally unrelated molecules like prostratin. we saw something similar when we tested this in vivo. So this is EAE model once again, in which we were treating mice in a therapeutic regimen. So we randomized and initiated treatment after they developed neurologic disability, and found that Bryostatin 1 attenuated disease. Bryo is are these triangles here, and this was replicated by certain bryologs like this one SUW133. And this effect was abrogated when you use derivatives that did not bind to protein kinase C. And so from here, this is kind of the lead in to the preprint. So particularly from my perspective, we have so many anti-inflammatory therapies for MS, but the big interest was that we have this a drug that was already known to be CNS penetrant in Bryostatin 1 that seems to target myeloid cells in the periphery. And so the question was, does Bryostatin 1 have similar favorable effects on myeloid cells within the central nervous system, specifically microglia and CNS-associated macrophages? And so that's the beginning of this story. And so I'm going to stop sharing my screen and turn it over to Paul to take over the rest.
Yeah, so as Michael mentioned, so we were interested in, we had this great data from, mostly, peripheral systems. So we were looking at macrophages and dendritic cells. So we're really more interested in the effects of it in the central nervous system. So, as a starting point, we cultured mixed glia culture, and we used mixed glia culture, which contains both microglia and which contains both microglia and astrocytes. So, maintaining that interaction, and so that we could see the effects of Bryo in this system. And from our previous work, we knew that Bryo 1 had anti-inflammatory, and so we treated mixed glia with LPS and conducted an RNA-seq analysis. And the RNA, this is your typical volcano plot showing that with Bryo 1 treatment, there's a lot of genes that are up and down regulated. And of those, when we looked at the pathways that were down regulated, we saw that the antigen, your classical pro-inflammatory pathways have been suppressed. So your antigen presenting process was decreased again. From our previous paper where we did the prophylactic treatment, that would suggest and why there was this suppression, but which we presume also occurs in the peripheral macrophages as well. But this is in the sort of the CNS mixed glia culture. So we have the decrease of NF-kappa B, TNF, NOD-like and Toll-like receptor signaling pathways, and the complement and cascade pathway. So this was, obviously really interesting to us because, we in MS, it's known that the complement system could play a significant role. One of them is the C1q protein, which is upregulated in microglia at the leading edges of the chronic active MS lesion. So, that was very interesting. And so when we looked at the suppression of this gene by Bryo, it was really interesting to us. And this is just a picture, a cartoon of how the complement system can activate microgliophagocytosis. And again, here's the C1q, and C3 is also very interesting because that there are variants of C3 that actually accelerates neurodegeneration in MS, and so that plays also a key role in the synaptic pruning and neurodegeneration. And we'll chat. I'll show some data about the C3 as well. So, and again, C3 is a marker for neurotoxic astrocytes, and so we were curious if Bryo also had an effect in astrocytes and their gene expression. And when you treat these mixed glia cells with Bryo, you see that there's suppression of these neurotoxic, your classical neurotoxic, some call them A1 astrocytes, while upregulating these neuroprotective genes. Again, here's the C3. And then we were curious, obviously this was mixed glia culture, so it contained both astrocytes and microglia. We wanted to know if there was a direct effect of Bryo on astrocytes. And we actually showed that it doesn't, and it's in the supplementary part of the manuscript. But when you take the conditioned media of just microglia treated with Bryo, you take that media and treat the astrocytes, then you see the decrease of those genes and increase of the neuroprotective genes. And similarly, you see just in general, like in these mixed glia cells, treatment with Bryo increases the expression of trophic factors, including BDNF and NGF, and suppressing the inflammatory pathway. And in our previous work, as Michael talked about earlier, we showed that Bryo also had this unique ability to potentiate the effects of IL-4. So have this, it was able to, I guess, express more of some of these, genes associated with the type 2 inflammatory response in macrophages. And we showed here that, again, it had similar effect in these mixed glia cultures. Again, increase in the the phagocytosis and cell cycle, and it increased gene expression again of those trophic factors and scavenger phagocytic genes, anti-inflammatory and proliferation. And so we saw this very profound effect on gene expression. And so naturally we were curious, does this translate into actual functional effect of Bryostatin? And so again, the first one, we just using primary microglia culture, we showed that when you treat microglia with Bryo, there's an increase of phagocytosis of myelin. And this is important, again, because myelin debris has been shown to inhibit remyelination. So one of the key functions of microglia is to induce phagocytosis and remove myelin, in addition to other harmful debris that are surrounding neurons. And in these panels, what we did was in wild type mice, we treated them with Bryo IP, injected them IP, the similar dosage and condition as the EAE conditions that Michael showed previously. We gave them 3 doses, so a week of Bryo treatment, and we extracted microglia and macrophages from the brain. So this shows that Bryo-1 can enter the CNS and activate microglia and macrophages to promote repair and regeneration. And so in these and then after extracting those cells, we incubated them with fluorescently labeled myelin for 4 hours and showed that indeed these cells isolated are primed to increase the phagocytosis of myelin, both in microglia and macrophages. And the other important thing is and what we saw from the RNA-seq data was that the Bryo-1 treatment released factors that could potentially promote oligodendrocyte differentiation and potentially remyelination. So in a cultured system, we isolated OPCs, the oligodendrocyte precursor cells, and obviously, just like the astrocytes, we treated them directly with Bryo, which again did not have any effect. But when you take microglia and treat them with Bryo and take that same conditioned media and put it in the OPC culture, we saw that there was an increase of oligodendrocytes as indicated by the OLIG2 and CC1 positive cells. And again, and F is actually showing the direct Bryo-1, and which you don't see that effect. And so now moving more towards the in vivo effects of Bryo. Again, we wanted to see if this was happening in mice. So we induced EAE, very similar to the previous EAE experiments. But at day 19, we started treating with Bryo, and then we isolated the brain and spinal cord on day 35 to isolate the microglia and macrophages to conduct RNA-seq. And what this showed again was that it downregulated your pro-inflammatory pathway, so the antigen processing and presentation pathway, TNF, and IL-6 productions, and so forth, while upregulating the regenerative pathways. So TGF beta, gliogenesis, and just differentiation and regeneration. And again, we talked about the importance of C1 and how in MS there's some variants of C3 that can accelerate neurodegeneration in MS patients. And in this case, we used TD tomato C3 reporter gene mice and induced EAE in these mice to see if Bryo can suppress C3 expression in astrocytes and microglia, which with Bryo-1 treatment, it suppressed. Again, kind of verifying our in vitro data in vivo system. And finally, we used another model of demyelination, which is the lysolecithin. You inject lysolecithin into the spinal cord, which causes demyelination. And on day 2, so we inject it to allow the demyelination to occur. And then on day 2, we start treating mice IP with Bryo. And at day 9, so about a week later and 2 weeks later, we examine if there's increase of remyelination. Typically, with lysolecithin, there's a spontaneous remyelination that occurs, and by day 21, like most of the remyelination have occurred, and so that's why we decided to look at some of these earlier time points of day 9 and day 15. And when we did that, one of the things that we saw was that there was again, because we, just like other experiments, Bryo-1 had an anti-inflammatory effect. So on day 9, you could see very clearly that it decreased the number of iNOS positive microglia. So iNOS is a marker for pro-inflammatory microglia, and so you see the suppression of that. And again, day 15, we don't see a statistical change difference, but however, when you look at the regenerative microglia, as indicated by Arch positive cells, Arch1 positive cells, at the beginning, there wasn't a statistical difference between the two mice, the mice that were treated with vehicle or Bryo, but by day 15, there's an increase of Arch1 positive cells. So this sort of indicates that, at the beginning, perhaps Bryo1's main job is to suppress the pro-inflammatory pathway or pro-inflammatory microglia, and then try to promote this anti-inflammatory regenerative phenotype in the CNS innate immune cells, and so this ultimately we believe sort of leads to increase of OPC differentiation and remyelination. So in day 9 and day 15, you see increase of OLAG2 positive, OLAG2 CC1 positive cells. And when we did qPCR, we saw increase of OLAG2, myelin basic protein and MAG, but mostly OLAG2 and MBP were elevated. MAG not elevated, but not statistically significant. And so when we stain them with MBP, you see an increase of MBP staining. When we conducted a EM, you see that Bryo1 enhanced remyelination as indicated by, or end maturation as indicated by the decrease of G ratio. And G ratio is really, the ratio of the radius of the inner circumference, like the cell up to the cell membrane to the end of the myelin. So I guess the thicker the myelination, you're going to have a smaller G ratio. And finally, we looked at both male and female mice, and seemed that both in male and female, there's Bryo1 enhances remyelination in both male and female. And finally, acknowledgements. So this is a group of people. Again, Michael and I, we've been working on this for a while. I know Michael said we've been working on it since PhD, but it wasn't that long. We've known each other since then in Saul Snyder's lab. But when we became faculty members here at Hopkins, we actually obviously knew each other from medical school, and became good friends, and we kind of knew each other throughout all these years. As I tell Michael, I probably knew him know him for half his lifetime at this point. But Payam is a postdoc in Michael's lab, and Efrat was a tech in Michael's lab, but is now at Yale. Sharuthi is a postdoc in my lab, and Wesley and Judy are graduate students in Michael's lab. And Matt Smith is, I believe he's a instructor now, I think, or something like that, in Peter Calabrese's lab. And we obviously collaborate with Peter and Jeff Huang at Georgetown on this work. And again, always, we've our initial EAE work started in Saul's lab because both Michael and I, we were stationed there when we were faculty members with using their lab space, and obviously our funding from DOD, MII, Race to Race MS. So, and that's it.
Excellent. Does anybody have any questions, or can I?
A question for Paul.
Yeah.
Hi, I'm Hayan. I'm from Genentech. This is my personal question, but just out of curiosity. So you showed that the Bryostatin treated mixed glia cell, you have upregulated markers of microglia.
Mm-hmm.
So is that the number of microglia, or is actually each? Is the gene per cell actually upregulated?
So that I guess if I'm understanding the question, you mean the RNA-seq data?
Yeah, yeah. In the in your bioarchive paper, there's a IL-4 Bryo treatment versus Bryo treatment. So it's I see all these microglia specific genes upregulated. So actually, my straightforward guess will be that you have extra like more microglia in the mixed glia culture after the treatment. So suggesting that maybe inhibiting PKC will actually promote the growth of these cells. Is that true or?
Yeah. Well, I guess we like to, consider Bryo as a PKC modulator. I know it has some inhibitory effects when you compare it to formyl ester, right? So it blocks some of the effects of formyl ester. But I guess we sometimes just kind of look at it as a modulator. But when we do that, right, it does increase the expression of these genes in microglia, and yeah.
So you haven't you didn't count the number of cells in the mixed culture. Okay. Yeah.
Right.
Thank you. Yeah.
Well, yeah. I mean, yes and no. I mean, we have done staining before and after, and with Bryo and without, and after the 24-hour treatment in mixed glial cultures, you don't see in fairness, we haven't looked exclusively at proliferation, but in terms of the comparative populations of like microglia versus astrocytes, we haven't seen a change there.
Right.
And it's not all upregulation of microglia genes. Some are up and some are down. In vitro, we have looked at proliferation,
Right.
and in the microglia, there's not an effect on isolated microglia cultures.
So it actually increased the expression of those. Yeah, I think that. Yeah, thank you so much.
Oh yeah, sorry.
Yeah,
There's an increase of those genes, but not they're not increased because there are more of the cells.
Right. So your red, like the most red color in that profile is like twofold, or is more than twofold change?
It's those are more I believe they're more than it's a log.
Yeah.
So yeah.
But there's no there's no
in the heat
number,
map,
so I cannot tell.
We don't show that. Yeah. I mean there's quite a range. Like for any one gene, I can't tell you. We can't tell you off the top of our head what the fold change is.
Yeah.
Have and I think it's loaded. I think it's in the supplemental material with the preprint. The actual like the raw data, like with the gene tables. So any particular gene you can look up and see what the fold change was.
Yeah. Thank you. Exciting. Yeah.
I want to ask a really simple question.
Sure.
Is the Bryo you're using isolated from the marine invertebrates, or are you synthesizing it, or are you going to use a homolog?
Yeah, so the ones that that's commercially available are the ones that are isolated. Paul Wender's group have like the synthetic version of Bryo 1. But yeah, so we have been using
that would be difficult to upscale.
It is, but he was able to do it. He's got grams of that.
He's doing it for EAE. I mean, now you're talking about one giving it to lots of people.
Yeah,
that's a lot more. That's a lot more stuff.
It is, but it's very potent. Bryo 1 is very potent. Yeah, and but yeah, for people we would need a lot, and obviously for those reasons, one, the marine source is not really easily scalable, and I think that's one of the reasons when we talked to Paul was to figure out, are there analogs and other simpler molecules that we could develop, and Bryostatin being one, and there are a couple of other PKC modulators that have very similar effects as
how would you You've done everything IV and IP, right?
Yeah.
How are you going to convert that to going to, as you move up to try and, because I have seen some people giving it orally. Have you ever looked at that?
We have. We haven't published it, but it works orally as well. You know, we've done one experiment, you know, doing the gastric lavage pathway, and it is orally bioavailable, and we have seen the effects. But we haven't done like dose to see the optimal. We kind of gave it the same amount.
No, I get you.
Yeah, I mean, what I would say is, you know, Bryostatin 1 itself has the advantage that it has been best studied. It has human safety data, but I think both of us kind of view Bryostatin 1, particularly, you know, Bryostatin 1 that is delivered you know, parenterally, you know, not orally, as kind of a proof of principle. And, you know, we understand that, like, in terms of what a future therapeutic might look like, you know, for a lot of reasons, it's probably not the most practical choice. And so, you know, that's why we are interested in formulations that would make it more orally available, and also, you know, as Paul mentioned, in synthetic bryologs that were easier to synthesize. You know, Paul Wender is kind of a dynamo, and he, you know, feels very strongly that they can produce enough of Bryostatin 1, and they're doing that for some clinical trials in leukemia. But you know, but like, clearly, you know, he also understands that you know, a drug that is simpler synthetically would be a big advantage.
Have you guys done anything in an E&E model that's relapsing, like, you know, PLP 139, 151 and SJL, or anything like that?
We haven't, no.
Because I have to say, you know, having been chair of CNBT and done EAE all my life, I mean, in many ways, there's a lot of advantages for the B6 MOG model, right? One, obviously, is that lots of people have knockouts and things in B6, but I make the argument it's not a good model for MS because it's not really relapsing. I'd make an argument, and I say this now, it's a model for MOGAD, which is a monophasic disorder directed against MOG.
Yeah,
And
no, I
that's
think that's fair.
Yeah.
Yeah, I think that's fair. And I also think, I mean,
it shows, I mean, let's face it, you show some important things that are probably true in either MS or MOGAD in terms of stimulating remyelination, right? The issue is going to end up being, you know, when push comes to shove, you need to probably be able to see when people did studies like this in Lewis rat or in MOG, the mechanisms that result in the fact that it's monophasic are very strong. And you need to make sure that those effects that you're seeing on cytokines, what happens when you have the conditions that allow for a true relapse to occur.
yeah, no,
But otherwise, it's, I mean, don't get me wrong, it's really interesting stuff. But having been in this game for a while.
Yeah, no, it is more than fair. You know, obviously EAE, I mean, a big part of the reason why, you know, we move towards a more pure remyelination model is because obviously, you know, many drugs have worked in EAE and then not had a benefit in MS. And even beyond that, you know, our big interests are beyond effects on you know, on even relapses. I mean, I feel like, you know, we're pretty good at that.
No, I mean, that 28 that 28-day data is in some sense, I mean, it'd be very interesting to know, like, what's happening pathologically there, because you're having an effect at a time when basically not in most people's hands, nothing is happening, and you're seeing recovery that's much better than what people typically see in that stage in 35-55 MOG-EAE. So, and I like the idea of the link of inflammation to subsequent remyelination. You know, the fact that you do the lysolecithin, and I don't know whether you guys have thought of also maybe doing cuprazone. There's other things obviously you can do to sort of try to differentiate how much of this is actually stimulating myelin versus stimulating immune cells that somehow then have an effect on remyelination. That's going to be, that's highly relevant, obviously, for the disorders you're looking at.
Yes, thank you.
Yeah.
Agreed.